In this video, we'll look at both a monocot flower and a Eudicot flower. In this first activity, we'll examine a lily, which is a classic example of a monocot flower. Lilies are ideal for studying basic floral structure because their parts are large, distinct, and clearly arranged in whorls. As you work through this activity, you'll analyze floral symmetry, count floral parts, determine ovary position, and identify reproductive structures. Since you'll be working from photographs, it's especially important to slow down and observe carefully. Before we begin observing the lily, let's review a few key evolutionary patterns in flowers. Monocots typically have floral parts in multiples of 3. Eudicots usually have parts in 4s or 5s. This reflects deep developmental differences in these two major angiosperm groups. Because monocots and eudicots diverged early in angiosperm evolution, and their floral organ numbers reflect genetic and developmental differences. You'll also evaluate symmetry. An actinomorphic flower has radial symmetry. It can be divided into mirror images along multiple planes. A zygomorphic flower has bilateral symmetry. Only one plane divides it evenly. Early flowers were genetically actinomorphic. Over time, some lineages evolved bilateral symmetry as an adaptation to specialized pollinators. As we move forward, remember patterns in floral number and symmetry often reflect evolutionary history. When you're observing flowers, one of the first structural features to look for is whether the petals and sepals are separate or fused. If the petals are separate, each one is distinct and individually attached to the receptacle. You can clearly see where one ends and the next begins. If they are fused, they are joined together at least at the base. Sometimes they form a tube, a funnel shape, or a bell shape. This is common in many advanced Eudicot flowers. The same applies to sepals. Separate sepals form a distinct calyx. Fused sepals form a united calyx. In botanical terms, fusion among similar parts is called conation. Evolutionarily, fusion often reflects specialization, especially for pollination. Tubular flowers, for example, often match specific pollinators like hummingbirds or moths. So when you look at the photos, don't just count the petals. Look closely at whether they are free or joined. That detail matters. Now let's review ovary position, because you will be asked to determine this in the lily. If the ovary sits above the attachment of sepals and petals, the flower is hypogynous, and the ovary is superior. If the ovary is embedded below the attachment of sepals and petals, the flower is epigynous, and the ovary is inferior. If it is partially surrounded, it is perigynous, or half inferior. You'll also need to recognize internal structure. The ovary contains ovules. Inside the ovary are chambers called locules. Each locule is formed by one or more carpels, which are modified leaves. When you look at the cross-section photo, you can use the number of locules to help determine the number of carpels. This is where structure tells you developmental origin. Now let's talk about pistils. A simple pistil is made from a single carpel. That means it typically has one ovary chamber, one locule, and one stigma. A compound pistil is made from multiple carpels that have fused together. You can often detect this by counting locules in a cross-section of the ovary. If you see 3 chambers, it suggests 3 fused carpels. Sometimes the stigma also gives it away. It may appear lobed or branched, reflecting the number of carpels involved. This is developmental anatomy. Carpels are modified leaves. When multiple carpels fuse, they create a compound structure. So when you examine ovary cross-sections in this lab, you are essentially reverse engineering how many carpels were involved in building that pistil. Structure reveals origin. Before we make any observations, pause and consider this question. Why would monocots consistently show floral parts in 3s? Think about developmental patterns, evolutionary lineage, and genetic regulation of floral organ formation. Write your prediction in your lab notebook before continuing. Let's begin with the external structure. First, determine the type of symmetry. Can the flower be divided into equal halves along multiple planes? If so, it is actinomorphic. or has radial symmetry. If it can only be divided in two halves, then it is zygomorphic or has bilateral symmetry. Now count the sepals and petals. Are they fused or separate? What color are they? Take a look at the less mature flowers. You see how some are green and others are pinkening? Those are the sepals. They turn the same shade as the petals inside. In this case, they actually refer to them as tepals when you can't distinguish between the two. So how many sepals are there and how many petals? You should have gotten 3 sepals and 3 petals, or 6 tepals. They are all separate. Notice the number of floral parts. Do they occur in multiples of 3? Now draw the entire flower in your lab notebook. Label the sepals and petals. Focus on structural accuracy, not artistic detail. Drawing forces you to slow down and truly observe. Pause the video while you complete your sketch. Now examine the longitudinal section. Count the number of stamens. I enlarged the photo from the previous slide. You can see how the stamens are turning inside out to reveal the pollen, called dehiscence. In the photo on the right, the anthers have been fully rolled back and the pollen is falling on the petals. Locate the pistil. Is there one? More than one? Examine the stigma on the left. Does it appear branched or lobed? Now determine ovary position by looking at the image on the right. The pistil has been split open. Look at where the petals and sepals attach relative to the ovary. Does the ovary sit above them or below them? Record whether the ovary is superior or inferior. Now draw this longitudinal view in your lab notebook. Label the stigma, style, ovary, and stamens. Indicate whether the pistil is simple or compound based on what you observe. Again, drawing is part of the analysis. Now examine the ovary cross-section. Count the number of locules. The chambers inside the ovary. You should see 3. Each locule responds to a carpel. This allows you to determine whether the pistil is composed of multiple fused carpels. Locate the ovules inside the chambers. Now draw the ovary cross-section in your lab notebook. Label the ovule, locule, and carpel. This is developmental evidence. You are using structure to infer origin. Pause the video and complete your drawing before moving on. To summarize the lily, it represents the monocot floral pattern, parts in multiples of 3. It is radially symmetrical. Its floral parts are generally separate. It has a superior ovary, and its pistil is compound, formed from multiple fused carpels. These features reflect monocot developmental and evolutionary patterns. In this activity, we'll examine hibiscus, a classic example of a Eudicot flower. Unlike the lily, which represents monocot structure, hibiscus shows us a different floral organization pattern. As you work through this activity, pay attention to floral symmetry, number of parts, fusion of structures, ovary position, and especially the staminal column. We're building a comparison in your mind between monocot and Eudicot floral architecture. Eudicots typically have floral parts in 4s or 5s, or multiples of 5s. They also often show more fusion of floral parts compared to many monocots. Fusion is frequently associated with evolutionary specialization, particularly for animal pollination. In hibiscus, we'll see an excellent example of fusion, especially in the stamens. As you observe, think about how fusion might improve pollen transfer. Before observing the flower closely, pause and consider this question. Why would a plant fuse many stamens into a single column around the style? How might that increase the efficiency of pollen transfer? Think about how a pollinator attracts physically with the flower. Write your prediction before continuing. Begin by examining the external structure. Determine the symmetry. Is it actinomorphic or zygomorphic? Count the sepals. Are they fused into a calyx? Count the petals. Are they separate or partially fused? What color are they? How many do you see? Notice whether the parts occur in multiples of 5, a hallmark of Eudicots. Now draw the entire flower in your lab notebook. Label the sepals and petals. Focus on structural clarity, not artistic perfection. Pause and complete your sketch. Now examine the reproductive structures. Notice the staminal column. Many stamens fuse together surrounding the style. This is a defining characteristic of hibiscus. Count the number of stigma lobes at the top of the style. Do you see any branching? That can indicate multiple fused carpels. Now determine ovary position. Does the ovary sit above the attachment of petals and sepals or below? Record whether it is superior or inferior. Now draw the reproductive structures in your notebook. Label the staminal column, stigma, style, and ovary. Indicate whether the pistil appears simple or compound based on stigma branching and ovary structure. Pause and complete your drawing. Now examine the ovary cross-section. Count the number of locules. How many chambers are visible? Each locule responds to a carpel. Locate the ovules inside each chamber. Now draw and label the ovule, locule, and carpel. You are using structure to determine how many carpels formed this pistil. Pause and complete your drawing. Now let's think more deeply. Compare the lily and hibiscus. How do the number of floral parts differ? Which shows multiples of 3? Which shows multiples of 5? Compare fusion. Did one show more fused structures? Compare ovary position. And finally, step back and ask, how do these differences reflect monocot versus Eudicot evolutionary patterns? Write your responses in your lab notebook using evidence from your observations. To summarize the hibiscus, it represents the Eudicot pattern, parts in multiples of 5. It shows more fusion, especially in the staminal column. It has a compound pistil. Its structure reflects specialization for animal pollination. When you compare lily and hibiscus, you're not just comparing two flowers. You're comparing two major evolutionary lineages of flowering plants. And once again, structure supports function.